Hi everyone, this is the Scrutinizer, aka Dr. Meenakshi Noll. Okay, we have vaccines against COVID-19. Pretty impressive that we have two that are released and more in the making. There has been a lot of confusion regarding how are these vaccines different than the classical ones, and I have had a lot of questions. So I wanted to explain all about the vaccines that have been released. These are valid questions that what side effects, if any, the new vaccines might have. So far, let's not forget that the classical vaccines have saved millions of lives every year and have successfully eradicated a lot of deadly diseases, a number of deadly diseases like smallpox, measles, polio, and other childhood diseases around the world. So very briefly, I would like to explain today the conventional vaccines, how do the vaccines work, and then to know that we need to know the functioning of our immune system, and I'll explain in short the functioning of the immune system. We'll go over the current vaccines, which are the mRNA vaccines, and the differences in the two vaccines that have been released in the market now or approved by FDA. They are from Moderna and Pfizer. And then, of course, the pros and the cons of the vaccines. And why do I say that we still need the phase 3 trial data to ensure the safety of these vaccines? Okay, coming to the conventional vaccines. There are four kinds. There are the four categories in this case. That the number one is they use the live or weakened altered organisms or pathogens, meaning that we use the pathogen but it is weakened, so some of the disease causing parts are taken out, so when they are administered in the humans they do not cause disease. And the example of such vaccine are the measles, mumps, and rubella. The second category is the inactivated bacteria or the virus. Third is the toxins, inactivated toxins. And the fourth is using the subunits. So coming to the second category where inactivated bacteria or the virus is used, it means that these are heat killed, means they are completely dead, but they can elicit an immune response in the body. Example of these vaccines are polio and Hep A. The third category is, like I said, inactivated toxins. What this means is that sometimes the bacteria itself is not causing the disease, but the toxins that are produced from the bacteria, they cause the disease. So in this case, we use the toxin to make the vaccine. Okay, the examples are diphtheria and tetanus. And lastly, the fourth category is that we can use just a segment of the pathogen, meaning either we can use the subunit or the conjugate vaccines. And the examples of these would be against Hep B, meningococcus, they cause meningitis, or the pneumococcus, which cause pneumonia. Okay, so how do these vaccines work? So when the germs, the bacteria or the virus, when they invade the body, they attack and multiply. We all know that by now. And this invasion is called infection. And what this invasion does to the body is called an illness. And we need vaccines to combat the illness. So what happens is that body tries to fight the intruders with various tools. So on the whole, the structure is called the immune system that is trying to fight off these intruders. So immune system, the functioning of the immune system, it is comprised of white blood cells. They are also called immune cells. So there are three kinds of immune cells or the white blood cells that will fight off the infection. The macrophages, the B lymphocytes, and T lymphocytes. And you must have heard a lot lately about the talk about B cells and the T cells. So these are the white blood cells. Macrophages, what are the macrophages? These are the white blood cells that swallow up and digest the germs. plus the dead or the dying cells. So they're gonna just ingest all of these things. But they also leave behind parts of these invading germs, which are called antigens, because anything that's foreign to the body is called an antigen, anything. And then the body identifies these, even they're dead parts of the microbe, but the body identifies them as antigen and that they are dangerous to the body. They stimulate antibodies to attack them. Then we have T lymphocytes, as you've heard, the T cells. These are defensive blood cells. They attack the cells in the body that have already been infected. Okay, and then we have another type of defensive white blood cells called B lymphocytes. B lymphocytes need the help of T lymphocytes to produce antibodies. And these are the antibodies, they attack the antigens, which are the foreign material left behind by the macrophages. And this B lymphocyte, when the B lymphocytes get involved, this is called the adaptive immune system. And in the T lymphocytes, it's called the innate immune system. So after the infection, the immune system remembers what it learned. Unlike us humans, we never remember from our mistakes. And the immune system remembers what it has learned and how to protect the body against the same disease or the intruder. And the body keeps a few T lymphocytes because they are the memory cells. And they go into action quickly if the body encounters the same germ or the microbe again. So when the familiar antigens are detected, the B lymphocytes, they produce antibodies to attack them. So why do we need vaccines if our body can fight infections, if we can do a good job on its own? And as we know, a lot of people lately do say we don't need vaccinations, I've got an immune system, or they refuse to follow the mandates, for example, the masks and the social distancing for COVID, because their reasoning being, I got an immune system, it can fight. So as we have seen in case of COVID-19, a lot of deaths, and then we also saw in case of smallpox, measles, the immune system was not sufficient to protect us. Why is that then? Because the first time the body encounters a germ or a microbe, it can take several days for the body to make all the tools, and I call them the soldiers, that are needed to fight the intruders. Secondly, the immune response that finally does develop after a few days, it may not be as robust as the body needs to fight off those viruses or the bacteria. Coming to how do the vaccines work? Now, the vaccines work by training the body to recognize and respond to the proteins produced by disease-causing organisms. And these, again, being the virus and the bacteria, or the toxoids. So basically, the vaccines are going to greatly reduce the risk of infection by working with body's natural defenses to safely develop the immunity to the disease. And what are the current vaccines, or which have been okayed by the FDA? These are against COVID-19. So these are the mRNA vaccine. mRNA, as the name says, M stands for messenger, RNA is the ribose nucleic acid, meaning this molecule has a ribose, which is a 5-carbon sugar, just like a fructose. Ribose is a 5-carbon sugar, and then there is a nucleic acid. I would like to discuss only the 2 vaccines that are released, that are from Pfizer and Moderna. In the subsequent videos, I will explain the other different kinds of vaccines that are in the making. For example, AstraZeneca is making a DNA vaccine. They are using an adenovirus. It's a recombinant viral vector. and they are encoding DNA in it. Then there is one from Oxford Research Center, and they have used the mixed approach, meaning they're using the adenovirus, and then they're adding the protein in it as the classical virus. So not to make too much confusion in this video, and I wanted to discuss the basics. Those vaccines I will discuss later on, but today we are going to focus on what are the mRNA vaccines. And as I said, as I discussed, the mRNA means messenger RNA, and it literally means a molecule that delivers a message. You can just call it as you would give it to a mail service or a human to deliver a message. So in contrast to all the above vaccines I discussed, that is the classical vaccines, the mRNA vaccines, they trick the body into producing some of the viral protein itself. Pretty ingenious, huh? In case of COVID-19, a synthetic version of mRNA is produced in the lab, which is similar to the original one that virus will use to build its infectious protein. Now, this mRNA has instructions on it to make parts of the spike protein, not the full-length spike protein, but parts of it. So it does not reassemble. So when this mRNA is sent in the body or introduced in the body, the cells think they are being asked to make the spike protein. That's what messenger RNA is delivering the message to our cells, saying make these parts of the spike protein. And the protein is still foreign to the body. So our body, because that is the message sent, our body will start creating those parts of the molecule that it is being asked. The immune system, however, will detect it, because for the immune system, these are foreign things. And so they will start producing a defensive response in response to these protein molecules. In case of classical vaccines, when I discussed above, they work only with the acquired immune system, meaning to activate the innate immune system, they need an adjuvant. We have been adding adjuvant that's called, for example, an alum. And I know that a lot of people don't understand the function of adjuvant, and they have had a lot of reservations against it. But an adjuvant is there. It just sits in there and it activates the innate immune system. But the good point about mRNA vaccines is that it can also trigger the innate immune system, but it does not need any adjuvant. So it does add, hopefully, it does add an extra layer of defense without the need to add the adjuvants. So in case of mRNA, all kinds of immune cells are being activated, meaning it's priming the immune system to get prepared for an endangering pathogen. In this case, it would be the spike protein or the coronavirus 19. And thus, the type of immune response that is triggered should be very strong, and should being the key word here, because we don't know that yet. And so how is the vaccine from Pfizer and Moderna different? Both of these companies are using almost the same technology. It is mRNA, and it is being sent in the body. It sits in a LPS. It is a lipopolysaccharide globule, meaning it's a little fat droplet, and they put the mRNA in it. When injected, the LPS, it gets absorbed into our cells, the muscle, and then the RNA is going to go further, getting into the cytoplasm and then do its work. But the difference was that Moderna is using a self-amplifying RNA, and Pfizer is using a non-replicating mRNA. And in both, or everyone is trying to do the best so that we have a robust response to the vaccine, and hopefully we can combat the COVID-19. Okay, the conventional mRNA vaccines, which is being used by Pfizer, the mRNA is engineered to resemble the full length or the fully processed mature mRNA molecule and encode only for the antigen of interest. Okay, in this case, it's the parts of the spike protein. So that is what Pfizer is doing, and otherwise they also are sending the mRNA in LPS. And then Moderna has used a self-amplifying RNA, and they do have mouse studies they have done in the past until last year, showing promising results, but this has never been done in the humans. It was never even given FDA's approval to be used in humans, so we still need a lot of research and phase 3 clinical trial data. Self-amplifying RNA, what does that mean? It means it not only encodes for the antigen, but also the viral replication machinery that enables the intracellular RNA to amplify and thus hopefully make abundant protein or express a lot of protein. In simple words, it means that the RNA that would go in, it will go into couple of rounds of replication. So it would make more RNA, and that RNA would make more protein. And that is the hope, that we would have a robust protein expression. Okay. And there is, as I said, a lot of work to be done to understand this response, the length of the protection it could give, and whether there are any downsides to all this. I will go over the beneficial features, because after all, this is a really great idea. It has been in the making for years. The only thing we don't have is we have never studied in the humans, and that's why we could be a bit skeptic. So the use of mRNA and over the classical vaccines that I already described, as well as the DNA-based vaccines that AstraZeneca is making. First of all is safety. So mRNA is pretty safe to say, meaning it is non-infectious. There is no potential risk of infection. It does not integrate in the body like the DNA does, and it cannot cause or introduce any mutations in the body. It just can't. Because one of the negative features of what we had to work in the past for mRNA is that it is degraded easily by the normal cellular processes. So it's in the body and it could get degraded really fast. So in a way, it is pretty safe, unlike the DNA. But we don't know how long the mRNA from both of these vaccines will stay in the body. But half-life, we can regulate the half-life. Once again, we need more research and more trial and error to do this, so know what kind of half-life we should have. Then we can also so as I explained earlier, that mRNA is inherently immunogenic. Its safety profile can be further increased. Once again, we can increase the safety profile, but this is the first time we have used it in the humans, so we need more research and long-term studies. Okay, the second to be discussed is efficacy. Various modifications can be made on these mRNA to make them more stable and highly translatable in the body. For example, right now we are using LPS, the lipopolysaccharide, as a carrier molecule, which is a little fat globule. And the efficient in vivo, which gives the efficient in vivo delivery. It also allows for the rapid uptake and expression in the cytoplasm. So mRNA is it's almost like no genetic vector, because like an adenovirus, which is a viral vector, of course it's safe, used in other vaccines, but mRNA cannot have any anti-vector immunity. So mRNA vaccine can be administered repeatedly. And then the third is the production of this vaccine. So mRNA vaccines have the potential for rapid, inexpensive, and scalable manufacturing because by getting in the human body to produce the viral protein itself, the RNA vaccines are cutting out a lot of the time of manufacturing processes that we would have to use. And so this should be easier and quicker to produce than the traditional vaccines. And we already saw that this vaccine came in the market for in a few months, which is pretty neat. And the quick and the manufacturing process, it's important if you think about deployment throughout the world and Europe and so forth. However, there could be the downsides. We have not had a chance to study these vaccines in humans because we have a pandemic at hand. This is an emergency situation. Otherwise, I'm pretty sure no vaccine is just released without a good phase 3 clinical trial data. So, for example, both Moderna and Pfizer, they have taken their control subjects or the endpoint. The way they took it, they can only say that the vaccine they produced is preventing the symptoms of the disease or reducing the symptoms of COVID infection. They cannot distinguish whether it is preventing the disease. Prevention should have been their goal, but their endpoint was to look for symptoms of the disease. Moderna's data, it comes from most of the recent clinical trials. They have mouse studies also. And in the clinical trial, the efficacy of virus was calculated to be 94%. And I would like to explain how did they do this, so you understand completely the whole clinical trial that they have done. Pfizer, I had a little difficulty obtaining all their data, but Moderna, I had it from their website also. So the total number of subjects that Moderna took was 30,000, and they split these 30,000 subjects evenly, that is 1-to-1 ratio, meaning 15,000 they took for the placebo group and 15,000 they administered the vaccine. Of course, it was a blinded study. We don't know who got what at the time. Now, the end point was once they see any of the subjects reporting symptoms, that's when they had to unblind the study. So out of all these 30,000 people, Moderna said that only 95 of the subjects, they reported COVID symptoms. And so when they unblinded the study to see the results, 90 subjects were found to be in the placebo group, and they had severe infection symptoms. And only 5 were from the vaccine group, and Moderna is saying that they had minimal symptoms. So from here, if we do the math, it does show 94% efficacy of the virus, and that the vaccine, all we can say from here is that the vaccine is reducing the symptoms. There's no way we can say it is protecting against infection, because we have asymptomatic people also, which either these companies they just forgot or they were in a hurry and they did not do the right controls. It would have been nice if they have spent so much money, time, and of course thanks to these 30,000 people and 43,000 for Pfizer that came forward to actually be the guinea pigs in a way, if I must say. And so if they had good controls, we would have had more answers from the same trial. Pfizer did say that it was too much to do, that is to screen the 43,000 subjects regularly as they should be, because if they were really looking whether it's preventing the disease or not, they the right way to do this clinical trial was to screen all the subjects for presence or absence of COVID before they were given the vaccine or the placebo, and then they are screened every week. So Pfizer stating that it was too much to do, no one said making a good, functional, and safe vaccine that actually protects or eradicates diseases like we have in the past. And doing the clinical trial was trivial. So I would say, a quote from my favorite game, which is chess, that strategy is an integral part of winning. Small mindedness and impatience for victory leads only to self demise and certain defeat. As we know that the companies were in a way competing, but maybe they were. Uh, I will give them benefit of doubt and say they were competing just for our interest for people and to eradicate COVID. But I strictly stick with the thing that strategy, we need to strategize even before we do an experiment. Okay, so from Pfizer's data and from what Pfizer released in the press release, they said that their clinical trials are showing that they have an efficacy of more than 90%. Now, in science and the data, it doesn't mean anything. It doesn't mean anything to me because I want numbers. If they are saying more than 90%, meaning they must have calculated it, then say so. Like Moderna said, 94%. So Pfizer needs to say percentage is a number, that it is that percent efficacy, not more than, not less than. That's not the way to correctly report the data, and then it makes me skeptic. Okay, secondly, both the companies are using, as I said, lipopolysaccharide as a vehicle to send the mRNA into the body. And Pfizer had stated that their control groups received saline as saline for the placebos. So their control groups received saline, which is not a good control, because if the mRNA is being administered with a lipid molecule, the good control is that the controls or the placebo group needs that amount of lipid as the control molecule, not saline, because LPS can also cause immunogenic effects, and we don't know whether the LPS is doing it or mRNA is doing it. So here also, I don't know if Moderna modified their trial later on, but this, at least in Pfizer, they had said that they used saline. So that was, again, using wrong controls. So I am emphasizing this just to show that there are such little tiny details that need to be addressed in a good study. Whether it's preclinical or clinical, we have to be careful about each and every little detail if we want to make a good vaccine, and also so that the people are not skeptic. And then the preclinical trial, what I mean is it's always the animal models. Mouse models so far, they have always translated the results very well so that we can go to the clinical trials. And Pfizer's data comes from large animals like macaques, monkeys. They are okay, but I am not in favor of using too many of the large animals. And how many can you even use? Like with the mouse model, they are small, and we can use at least 6, 12 at a time and have a good statistical data. Okay, coming to the fact that we need long-term data for the current vaccines. This is because, like I said, we do not know anything about mRNA eliciting an immune response in the body, in the humans. What we do know about mRNA in the human body, it comes from the work on cancer. The technology is really great for the oncology field because we can develop specific vaccines for different tumors and they are different in different people. So how does this work in tumors and what do we know? In tumor, we can use the tumor mRNA the same way as I discussed for the vaccine, that it can that the tumor RNA can stimulate people's immune response to recognize and then respond to the proteins produced by the specific tumors. And what does this do? It's like magic. It activates body's T cells and the part of the acquired immune system that kills cells, which is useful to destroy the tumor. So the same T cell response could be very important for coronavirus, but we don't know that yet. Why is that? What is the extra we need to study that we don't have from the tumor data? For a virus like coronavirus, we would need a strong T cell response. And why is that? Because not only the viruses are so sneaky that they take our body over, they go and hide in the cells. So the T cells are the ones that can go after them in various cells and devour the infected and the abnormal cells. So there is a certain hope, especially in this setting, that the mRNA technology could really work and eliminate the infected cells from the body. However, to fight such a virus like SARS-CoV-2, we need the B cells to be activated as well. And B cells, as I explained earlier, they are the ones that produce the antibodies that mark the virus out for destruction by the body. And this is where we have little or we don't have any experience from the tumor data, because in oncology, we're not looking for the B cells. We were only looking for T cells, and they elicited the immune response and thus killed the tumors. And in nutshell, there are a lot of unknowns that may or may not have a downside, because theoretically there are a lot of questions, and these are good questions if anyone asks, and they should have the answers. But to actually say whether there are any side effects or not, we will have to follow the phase 3 data long term. So what are the questions? Number one, we need to understand whether these vaccines will really be able to mount a sufficiently protective immune response in the humans. Then second is to understand what quantities of mRNA will be needed to do this. Right now, they have taken two arbitrary values, and the second injection is when some of the subjects did have issue, like the allergic response. So we still need to standardize the quantity of mRNA that will be needed. Number three is whether the protein that they have chosen for the vaccine, these are the right ones to prevent a coronavirus infection in the body. We chose the spike protein because that is the protein with which the virus makes entry into the body and attaches to our ACL receptor. Okay, so the fourth is whether the vaccine causes side effects such as increased inflammatory response like redness and swelling, or there could be worst case, whether it aggravates the disease. The extracellular naked RNA has been shown to increase the permeability of tightly packed endothelial cells. What this means is that it could cause edema. We have seen it in a couple of cases in the clinical trials where edema has happened, the swelling of the injection site. Okay, the next question would be, how targeted is the immune response to this particular coronavirus? Meaning, how long any immunity would last, if any. Both the companies, they have reported that there was a robust immune response. Now, if it is robust, first of all, it should prevent infection, which is not doing, or we cannot tell. But the second question would be, how long is that lasting? And they can only tell us by following their subjects. Okay, so next is that in vitro transcribed RNA. This is the mRNA that we're using for the vaccine, and this is inherently immunogenic, meaning it would contain contaminants like double-stranded RNA. And of course, before they're used in the vaccine, the whole thing is purified. HPLC is used and so forth. However, it's impossible um, to remove all the contaminants. And this could be the contaminants could be responsible for the innate immune activation. What it means is the response may not be specific, or there are recent human trials have demonstrated that moderate to severe injection site or the systemic reactions of for different mRNA platforms. Now, even if we have used the purified mRNA in the self-activating mRNA like Moderna is using, there would be the RNA is making more RNA. So every time it replicates, there would be production of double-stranded RNA. And so that double-stranded RNA would elicit an inflammatory response. And the mRNA is known to induce high levels of cytokine secretions, once again, immunogenic. So we still need to evaluate in future the preclinical and clinical studies, and these include local and systemic inflammation, the biodistribution, persistence of the expressed immunogen, stimulation of the autoreactive antibodies, and potential toxic effects of any non-native nucleotides and the delivery system components. This is LPS and mRNA. So the another thing to look for from the Moderna vaccine is, because it's using the self-activating mRNA, it is making double-stranded RNA that I said. These double-stranded RNAs, it is a potent pathogen-associated molecular pattern, PAMP. What does this mean? It means that there are conserved molecular structures produced by microorganisms, and they are recognized as danger signals by the innate receptors. So our various innate immune receptors, they look at them as foreign, and so they are gonna induce an inflammatory response to protect the cell or the body. And LPS is famous for generating that kind of response, and we have already seen in a few subjects or the other some of the people who received this vaccine, they did get an anaphylactic shock and anaphylactic response and so forth. However, having an allergic reaction, anaphylactic shock is serious, but having an allergic reaction is not on top of the list of concerns, because we can deal with it with antihistamines and so forth. But a possible concern with some of the mRNA-based vaccine platforms would be induction of potent type 1 interferon response, which have been associated not only with inflammation that we already discussed, but also with autoimmunity. So we would need to identify individuals who are at an increased risk of autoimmune reaction before this mRNA vaccine should be administered to them. This would be a reasonable precaution. Safety. So this all points towards safety, that we need continued evaluation as different mRNA modalities and delivery systems are utilized for the first time in humans and tested in larger patient population. So if I were to sum it up, I would say that the frontline workers, or the clinicians or the other people working in the ER, or who come in contact with the patients, and especially the COVID patients, they should definitely take the vaccine. And for others who are not coming in contact with patients, they are following the mandates so they don't get sick, they could wait for the long-term study. I say this because if the vaccine had shown that it is preventing the disease, of course, I'm all for it. Everyone should get vaccinated and we can say goodbye to coronavirus forever, hopefully. But as the data is showing, the two vaccines, they show great potential, but they are saying so far that they can only prevent the symptoms, not the disease. So this means that the vaccinated people, even if they get sick, they would not know that they're ill, and they would have a false sense of being healthy. However, they would be shedding the virus and transmitting the disease. So in a way, we are not eradicating the disease. So it seems that waiting for more of the data from Moderna and Pfizer would be a good thing to do. And there is more clinical data from Moderna, and their data seems to be cleaner than Pfizer. And so I say this that in my life I have lived, I've loved, I've lost, I've missed, I've trusted, I've made mistakes, but most of all I've learned. So what I'm saying is I'm laying a lot of stress on the safety of the people. Yes, we all want coronavirus gone. Yes, we all want a vaccine that works. However, wanting and having, and the vaccine actually delivering what we wanted to deliver, are very different things. So I lay stress on taking a moment, and that all of you should really look at the facts I provided, the facts you might already have and accumulated from wherever, and then make a good decision for yourself and your families. Of course, that decision has to be based on science, not any loyalties or friendships, because as I always say, the facts are the constant. Okay, loyalties and everything else change, and you want to do what's best for you and your family. So take a moment, cool headedly look at the data, and if you need to ask me, I'm always here. Send a message or write in the comments. I always reply, and I will always help deciphering the data. I'm not making decisions for anyone because my role is to provide the good facts. Okay, so in the end, as I always say, think critically, stay healthy, beat the COVID-19 with science. The Scrutinizer out.